Good morning, everybody. We're going to continue to cover adult stem cells. In the previous two lectures, we covered the hematopoietic stem cell and the mesenchymal stem cell mainly, and we also, spent some time on the discussion of satellite cells, which is the adult stem cell within skeletal muscles. So today we're going to describe uh, stem cells in the small intestine as well as in the colon. So the stem cells present in the small intestine is one of the best characterized, best studied, along with hematopoietic stem cells. In some sense, we can say that the adult stem cells in the small intestine was even better characterized than the hematopoietic stem cells, especially how how the niche cells interact with the stem cell. So let's start today's lecture, and I'm going to first describe uh, endoderm-derived organs. You know, uh, earlier we described that during early development, you first have a fertilized egg, and then after a few steps, you will reach the stage of blastocyst. That's when you can still take out the cell and keep the cells in the state of pluripotency. In blastocyst, the cells are almost equal to each other. You have quite a few cells, but those cells almost equal to each other because each one can derive into a new individual. So they are pluripotent. They can give rise to all the cell types theoretically. And then later on, the developing embryo will go through another process called gastrulation. The gastrulation is a process describing the formation of the three germ layers. The three germ layers are endoderm, mesoderm, and ectoderm. So today, we're going to discuss the small intestine, as well as the majority of the rest of the GI tract is from the endoderm. So let's start from there. I will describe the endoderm-derived organs. So first of all, it's the GI tract. In the GI tract, you have esophagus, and then followed by stomach, and then small intestine, and then it's going to be colon. So this is the tract of the digestion system. In addition to that, you have some other organs associated with the GI tract. Their function are closely related to digestion and nutrient absorption. So those organs are liver, gallbladder, and pancreas. So those, is not part of the GI tract, but they're closely associated with that, such as liver. The liver will filter out the toxin within the newly absorbed nutrient, right? The blood supply is from the portal vein. The portal vein will collect the nutrients absorbed from the small intestine, and then in the liver, it's going to complete the process of detoxing, right? And also the liver will secrete a lot of enzymes and in the bile, and the gallbladder will collect the bile and then transfer the bile into the small intestine. And then the pancreas, of course, it's a source of enzyme, the digestion enzyme that need to be transported into the small intestine as well. So they are closely associated with the GI tract. Now, all these organs in the GI tract, they're from endoderm. But you may not. That is probably easy to understand, and you remember that sometimes we call the GI tract the gut, right? It's your gut feeling. The gut is also a term we use to describe endoderm. So we call the endoderm foregut, midgut, and the hindgut. dividing the endoderm to 3 parts based on their anatomical position. So endoderm is kind of, you can call it gut. So the GI tract is easily understood that it is part of the endoderm. However, you may not know that the majority of the respiration system are also derived from endoderm. So here on B, I show you a picture of the respiration system. They have nasal cavity, pharynx, and then followed by larynx, and then trachea, and then you have the 2 branches. So if you further observe the finer structure, to the terminal of the lung, you will have a alveolar duct, and so-called alveoli, alveola. So that's the place where the oxygen will exchange with with the blood within the alveola. So the majority of the respiration system are also from the endoderm, except some part, like the nasal part and the pharynx. The nasal and pharynx, they are not from endoderm. Only beginning from larynx and then to the alveola, those are from endoderm. And then for the majority of the GI tract, as well as the respiration system. So when we say the cells are derived from endoderm, it is mainly the epithelial lining of these organs are from the endoderm. So because each organ of our body is very complex. They have blood vessels and a mesenchymal content. So like the blood vessels shown here, the pulmonary artery and the pulmonary vein, they are not from endoderm. So all the vein and the blood cells within, they're from mesoderm. Okay, the mesoderm gave rise to the circulation system, as well as the endothelial cells lining the vessels. So the epithelial lining of the GI tract and the respiration system, they are from the endoderm. Okay, so just to clarify that concept. So we say, okay, kidney is mainly from the mesoderm. The hematopoietic stem cell, hematopoietic system is mainly from mesoderm. And then GI tract respiration system is from the endoderm, but that's I would say it's a raw summary. You do have other germ layers that contribute to almost every organ throughout our body. Yes. Give me a minute. My screen is not moving. I don't know what's going on. Okay. Okay, here I'm going to give you a brief overview of development. So, my lab works in embryonic development. So this is something we do, regularly. So we just put a male and a female together. After a few days, we start to isolate the embryos for our purpose, for our study. So at E7.5, which is the date when you kind of 7 days after you put a female and a male together, we count that, we count the night as half a day. So we say that E7.5 is, the 7th day after you put the animals together. So E7.5 means maybe the embryo is now 7.5 days old, right? So at E7.5, what you're going to see is a cylinder-like structure for the embryo. It's already an individual. At this stage, the endoderm is the part, showing with a blue with a yellow color, which is basically, if you can see my the arrow on my mouse, that's like this. That's the outside layer, here of the bottom part of that structure. The outside layer of the bottom part is the endoderm at this stage. And then at E8.5, it's only 1 day after E7.5, you can tell that there is huge change there morphologically. So that's how fast the things work in embryo. In adult, so we grow for a year and then nothing will happen. We look still quite the same, but in embryo, the things happen really, really rapidly. So at this stage, you can see the head and the tail, but the head and tail are kind of far apart from each other. You see a strange shape. That's what happens at E8.5. There is a tract there, labeled by the yellow color again, but the yellow color won't justify some of the part. But you should appreciate it. It's a tract there. Okay, the tract at this point is called gut. But as I said, this is the process of endoderm patterning, and then you can divide that endoderm at this point to foregut, midgut, and hindgut. Okay, 3 parts of the gut. And then just 1 more day later, as you can see, huge change again. Now the tail bent forward. The tail bent forward, now getting closer to the head region. So that's pretty much the position that fetus will take, during the course of the embryonic development. So now the tract is getting clearer. You do see a tract there, but along the tract there will be some bud formed outside of that tract. Those are called organ bud. The organ bud will give rise to liver, pancreas, and gallbladder, because those are not within the tract. They're associated with the tract. So those, as shown in the middle region, they are called organ bud. They're going to come out. And then please don't pay too much attention to the color code, because somehow it is confusing. Like at the bottom of this figure, you see the red color, yellow color, but only the yellow color is consistent with what's shown, on top of that illustration. Well, the other color, the red color, doesn't really correspond to the same thing as what's shown on the risk part of that illustration. So here at the bottom, what you're seeing is a section at the place where I put my arrow there. So you do a section there, and this is what you're going to see. And in this section, in the middle is the gut. In the middle, it's a tract there. We shouldn't say it's GI tract, because at this point it's not the mature GI tract yet. It's just the gut. Well, surrounding the gut, the next is mesoderm. So the mesoderm will give rise to the body wall. Like in your body wall, you have a lot of muscles, right? So it's going to be the muscle of the body wall mainly. Of course, there are going to be additional structures there, but here we're talking about the layers of that fetus body. So the mesoderm is outside of the gut. Well, in the exterior region, you see the blue color. The blue color is the epidermis. They're from an ectoderm. Well as this, you see this ring here. This ring is called a neurotube. The neurotube will give rise to the spinal cord later. So that's the 3 germ layers in this section. Now the ectoderm now is on the outside. The mesoderm is in between. Well, the endoderm is it's the internal part. So now, based on the name, It's easy to memorize. Ectoderm outside, endoderm inside, mesoderm is in between. But at the beginning of development, the endoderm is actually the outside part. Okay, at the very beginning, the endoderm is on the surface of that embryo, at that E7.5 embryo. Okay, so here in B, it's just, I would describe that again, but we won't refer to those pictures. So the embryos at this stage, at the stage of forming 3 germ layers, is called gastrulate. The process is called gastrulation. So sometimes we say, although we refer the 3 germ layers as mesoderm, endoderm, and ectoderm, but a lot of times, when the germ layers branch out, first what you're going to see is ectoderm and mesendoderm. So mesendoderm is the common progenitor of mesoderm and endoderm. So in other words, mesoderm and endoderm, they are more closely associated with each other. So the ectoderm will branch out first, while the other 2 germ layers will branch out a little bit later. Okay, so the endoderm, as I said, have 4 parts: foregut, midgut, and hindgut. And now let's try to memorize what this part will become eventually. So the hindgut is very easy. It's the back of the end of the gut. It's going to give rise to colon and some of the urogenital organs. Okay, so colon and some part of the urogenital organs. And midgut is small intestine. Well, the foregut is the place you're going to see most of our critical organs throughout our body, like the upper part of the GI tract, esophagus, and stomach. Okay, those are all from the foregut, but the associated organs like liver, gallbladder, and pancreas, they're also from the foregut, and as well as the respiration system, the lung, trachea, okay, larynx, and so on. But something you might want to pay attention to is thyroid. The thyroid is a part of the endoderm as well. So that's, my description of endoderm-derived organs. As you can tell at this point, when we discuss stem cells, we cannot avoid discussing development because, they are so closely associated with each other, especially the pluripotent stem cells. So here, as I said today, we're not going to discuss all the adult stem cells within the GI tract and in the lung. I'm going to focus on the adult stem cells in the small intestine because it is one of the best characterized, perhaps the best characterized, and not just one of those. So one of the reasons why this is so well studied, so well characterized, is because, like the hematopoietic system, you have a lot of cell renewal here. So we put it to numbers. There are about 10 to the power of 11 cells lost every day. So this rate is very comparable to the cells in circulation, approximately the same number, right? Previously, I recall when we discussed the hematopoietic system, the renewal rate is about 10 to the power of 11 to 12, which is in line with the renewal rate of the epithelial cells in the small intestine, in the small and the large intestine. So in other words, we can say approximately every 3 to 5 days, all the cells in the epithelial lining of the small intestine will get completely replaced. Okay, so that's a high rate of cell replacement. So I would like to describe the structure of the small intestine because without that we cannot understand how the adult stem cells work. So in the function of the small intestine is mainly absorbing nutrient, right? The food is the food is already pretty processed by the stomach, and at this point it's more about absorbing the nutrition out of the processed mix there. So to serve that purpose, so the epithelial lining of the small intestine is trying to maximize the surface area, because when you have a lot of surface area, you have a lot of cell in contact with the outside, with the lumen. So to serve the purpose very well, you need to maximize the surface area of that epithelial lining. So now that's why you have, if you observe, you will see that there are presence of finger-like protrusions. So like shown in A, this is finger-like protrusions, and this is called villus. The plural form of that is called villi, but the villus is the unit there, which is about 0.5 to 1.6 millimeter long. It's quite long. That has really expanded the surface area. Under those villus, actually I should say at the bottom of the villus and around the base, this is, you have to imagine this is like a finger, just like a finger. At the base of that villus and at the base and surrounding the base of that villus, you're going to see structure that's called crypt. I couldn't pronounce the full name of it. I'll try. So there are at least 6 crypts of Lieberkühn. I would say that. I don't know whether it is correct. So at least 6 of them. Okay. So within this crypt, you're going to have cells generated. So the cells will get generated within the crypt, and then the cells will move up until they reach the tip of that villus. Okay, so if somebody said that the cell renewal here is because of the presence of stem cells, well, I think it's easy to imagine maybe the stem cells will be in that crypt rather than in the villus, right? Because the villus is performing the function, while the crypt is hidden or buried deep under that villus. You would think maybe the stem cells will be in that crypt. That's a straightforward thinking. Okay, so I'm going to describe the structure of the crypt a little bit. So in the next slide, we can describe what cells are the stem cells. So here we have the enterocyte that's absorptive. They are going to pick nutrients out of the lumen, right? And then you have this orange colored cell, which is the progenitor cells. So those cells are also called transient amplifying cells. So they have they're highly proliferative. So while they differentiate, they also amplify the number of cells. Okay, you have additional cell types along the length of the crypt, and some cells are called goblet. The goblet cells secrete mucus into the lumen. Okay, that's the function of goblet cells. You have tuft cells. The tuft cells is the sensory cell, that the chemosensors. Okay, and you also have enteroendocrine cells. By the name, they secrete hormones. And then you have M cell. M cell is a special cell types that mediate the interaction between the environment in the lumen and the immune cells underneath, the adjacent to the M cell. So this M cell is important for immune reaction. So you don't have to pay attention to these special cell types, because for the cell replacement, it's mainly about the absorptive and enterocyte. But of course, the other cell types are generated from the stem cell as well. So within the the bottom, the base of that crypt, you have two cell types. So here we show one is called a Paneth cell, but the other is a cell type that later, but people think is the adult stem cell there. Okay. So on this slide, I described two theories or two models that over time scientists has proposed to represent how cells are renewed within the crypt. So the first is called the +4 position model, and this model is mainly proposed by this scientist Chris Paulton, and who's considered the pioneer in adult stem cell research within the small intestine. So in his theory, so basically he said that at this +4 position that you will have stem cells. What is the +4 position? So basically you count from the bottom of that crypt, and then I don't know why he could call it +4, because if I count by this illustration, it should be +5, but it doesn't matter. At this point, the +4 becomes a scientific term. This is not really you count. It's just it says between the Paneth cell and the transient amplifying cell, you have this region, and in this region is very close to the bottom, but not exactly the bottom. It's close to the bottom, but not the very tip of that bottom. So you have about 16 cells that are considered stem cells. Why I say 16? Because remember, this is 3D, right? You have a tube here. So here, although you only see two cells, but remember, this is a tube. Along the circumstance of that tube, you have collectively 16 cells at that position, at that so-called +4 position. So in his theory, this +4 cell will give rise to the functional cells such as the enteral cell, goblet cell, endocrine cell, but it also should give rise to the Paneth cell, because the Paneth cell is considered to be the niche cell. The niche cell will provide the signal that will maintain the stem cell in their multipotent quiescent status. Okay, so that's the first model there. And later on in 1974, two scientists, Hazel Cheng and Charles Leblanc, proposed another model called stem cell zone model. So in that model, they consider that at the bottom of the crypt, you have not only the Paneth cell, but a stem cell which is morphologically distinctive from the Paneth cell. Okay, so they say this is the stem cell permissive zone. So this zone at the bottom of the crypt is responsible for producing, all the cells in the lining of the epithelium. So they didn't say at which position. They say collectively, the Paneth cell and another cell type will interact and eventually give rise to new cells that can replace other cell types within the lining of the small intestine. But they describe that at some place, that place is quite similar to the place of the +4 position. They in their model, they say at position +5 of the crypt base, you have a common origin of differentiation. Basically, they say around that +4 place, and now you set a boundary of differentiated cell and the adult stem cell. So when you move out of that +4 position, now differentiation will start. If you go below that position, the cells are just multipotent. They just self-renew. They don't differentiate. So they define that as common origin of differentiation. And in their model, they say they named this cell, this adult stem cell, as a crypt base columnar cell. Columnar describes the shape of it. is columnar. So it's a crypt-based columnar cell. Just as the first model, this cell will give rise to the functional cell as well as the dependent cell. But the dependent cell, it is placed in a very unique position toward the CBC cell. They're kind of intercalated with each other. It's not like intercalated. That means you have one dependent cell and the next is the stem cell. Then you have another dependent cell. That's what they propose to be the structure of the crypt base, as well as the mechanism governing the cell renewal in the epithelial lining. So what's their evidence for the second model? So their evidence is firstly electron microscopy. So they observed the shape of pennant cell at the bottom of that crypt, the crypt base, but they see there is some other cells that don't have the typical shape morphology of the pennant cell. So that's first evidence. There is other cell types there. And then in another experiment, what they observed is when they have tritiated thymidine, tritiated thymidine put into the crypt. So the tritiated thymidine is an isotope. It's 3H, it's 3H thymidine. So it's an isotope. When they incorporate that isotope to the base of the crypt, they kill a lot of cells because it's kind of irradiation there. So a lot of cells, the columnar cells, are killed, but some columnar cells survived. And then they observed morphologically that those survived cells kind of using phagosomes. Phagosomes is engulfing, right, of the neighboring dead cells. So the live cells engulfed the dead cells, but eventually, because that's radioisotope, right? So once they engulf other cells, they get labeled as well. Maybe at the beginning, they didn't receive a lot of the irradiation. They didn't incorporate a lot of the tritiated thymidine. But now, once they engulfed the dead cell, they are more labeled. They have more 3H thymidine in there. But amazingly, later on, they found that almost the entire lining of that region, of that small intestine, now become radioisotope labeled. What does that mean? That means the columnar cell eventually repopulate the surface of the epithelium of the small intestine. So that kind of demonstrates that the columnar cells have stem cell potential. So that's kind of labeling. Okay, the scientists are always relying on some kind of labeling to tell them a story. Okay, and later on, that is much later, it's 2 decades later. Hazel Chen, again, at the beginning was a trainee of the scientist, and now he become a professor. He and his colleagues now use the heritable somatic mutations to demonstrate the same point. Recall, what the presentation on Friday? The scientists used some kind of mutation induced by radioisotope to observe the single cell origin, the single cell origin of hematopoietic stem cells. Then here, so when you read this heritable somatic mutation, that should ring a bell. It tells you that these scientists are using very similar technique. They have some special somatic mutation they observed that happened in some of the columnar cells, and now they allow the animal to get older a few days. A few days later, when you dissect that section of the small intestine, now a lot more cells, the epithelial cells of the lining, now also bear the somatic mutation. So that means the columnar cells now give rise to more to the functional cells or the epithelial lining of the small intestine. So this is another evidence showing that the cells has potential, stem cell potential. So yes, please.
So basically, what you're saying is that the technique that was used by Tiller and by Till and the other scientists on that paper for the hematopoietic stem cells, it's used also for these kind of experiments as well. So it's like consistent throughout the decades.
No, I'm not saying that, because in this case, you know, maybe the somatic cell mutation is spontaneous. It's not induced by irradiation. Here is, you know, the mutation happens all the time, especially in on our epidermis and in the small intestine, because you're always exposed to certain kind of toxin.
Oh, okay.
Yeah, okay. So some mutation will be there. If you're careful enough, you can track that mutation.
Oh, that's cool. Thank you.
Yeah, this is just being creative.
Yeah.
Right? The scientists, whenever they see a chance to help them to accomplish their intended task, they just come up with the idea.
Yeah, really nice.
Okay. Yeah, you remember that's also quite early, although it's 1990s, but it's still quite early days of scientific research. So on this slide, I'm going to show you there's two scientists who are considered pioneers of this field. One is Poulton, who's the one who proposed the first model. The other is Hans Clevers. So Hans Clevers is a professor, from Holland, and he got his. At the beginning, he thought he would like to do biology. He get into a PhD program, but just a little bit later, he found maybe he wanted to be a doctor, and he also get himself into an MD program, and then somehow he couldn't make a decision. He did two projects, so one for his MD, one for his PhD. So he graduated as an MD first, and a year later he graduated as a PhD. It's not like nowadays you have an MD PhD. He graduated, at almost the same time with two different degrees. And then he found that he didn't want to be a doctor. He thought he's impatient to patients. So he moved on to pursue a research career. He was a postdoc in Harvard. And then, he made a lot of contributions from that point on. His major contribution is the identification of LGR5, which is the marker of the adult stem cells in the small intestine. But in my opinion, his other contributions, which is the description of the Wnt pathway, is even more influential, because at this stage, the stem cell is still at the early stage. Stem cell research is still at the early stage, so we don't yet have therapeutic use of his knowledge, his contributions, but his description of the Wnt pathway is applicable to quite a lot of the cancers, like the colon cancer. And so his contribution in delineating the pathway, so that how Wnt will interact with the receptor and how that will lead to the stabilization of beta-catenin, and beta-catenin now move into the nucleus to regulate gene expression, is some of his major contributions. And he's one of the most established, the most famous scientists from Netherlands. And I believe he's still quite young, and he's still making a lot of contributions. So I'm going to describe not his work. We're going to have a student-led discussion this Friday. I picked a paper from Hans Clevers, and we're going to have group 4. Can somebody from group 4 acknowledge that you're here? Group 4. I guess not a lot of listening. Anyway, so I posted the material for this Friday's presentation. I just want you to know that. If you don't, well, the materials are there. Okay, so here on this slide, I'm going to tell you something about linear tracking. So that's the definite evidence of the presence of stem cell and how the stem cell give rise to the rest of the cells in the epithelial lining of the small intestine. So that is based on the discovery by Hans Clevers that LGR5, which is a Wnt regulated gene and which is also a part of the Wnt pathway, is a marker of the stem cells. So once you know the stem cell has a marker, now you can track it using modern genetic approach. So in the figure, what is shown is so-called a linear tracking. But I want to point out when scientists have a marker, a specific marker, which is a genetic marker, they usually have two ways to track the daughter cells. So one way is to put an indicator or a reporter like a GFP directly under the control of the promoter of LGR5. So if you take that approach, basically the GFP or any other reporter that you put into the locus will have high fidelity to the original protein, which is LGR5. However, often that labeling is transient because when the cells differentiate, right? At the stem cell stage, they express LGR5, but once the cell will become the epithelial cell, they don't express LGR5 any longer. So you can imagine the ultimately differentiated cell will not have that labeling, right? But you can do a linear tracking. In a linear tracking, what you do is put a Cre, the recombinase to the locus of LGR5. Well, in another locus, you put the indicator, like you put an indicator in ROSA 26. So within ROSA, that reporter, within the ROSA locus, that reporter is not expressed because in that locus, you have a stop signal in front of that coding gene there. So only when Cre remove that stop signal, the indicator is now activated, it's now expressed. So this way, because the Cre-mediated recombination is irreversible. So although later on the LGR5 is not expressed, but it's the descendant cells, the progeny is permanently labeled. So this is a very important concept. I emphasized in my discussion of manipulating mouse embryo. I guarantee you I'm going to test that. I'm going to test that heavily in our final examination. So that's one of the most important content I want to teach in this class. If you don't know this concept, if you feel what I'm saying right now sounds so strange, you don't have any idea of what I'm talking about, better study this part because, as I said, it's going to be heavily tested in the final examination. So here, what they show using a linear tracking is now you have LGR5, Cre crossed with the ROSA 26 beta gal. So that when you cross these two animals, you can label the cells, the LGR5 positive cells, and its progeny, its daughter cells, daughter cells of daughter cells, all the cells, resulted from that one positive cell, permanently labeled by beta galactosidase positivity. And now you can see at 12 hours of that labeling, you see some cells at the base of the crypt, only small amount of cells. I would say like 2 or 3 cells there. And you can even distinguish that those cells are not continuous. That means the cells are not against each other. There is some space in between them, okay? So this kind of tell you there are multiple cell types within that plate, within that base. And then after 5 days, you can see the positive cells, the beta galactosidase positive cells are moving along that crypt, moving up. And then after 60 days, you can see those cells now reach the tip of the villus. So this clearly demonstrated that the stem cells within the base is now contributing to the entire length of the epithelium lining there. Okay, that's the final say, the final evidence, ultimate evidence to show indeed there's stem cells at the bottom of that crypt. And then this is from the paper that the student will present this Friday. So basically, Hans Clevers was able to show that when he isolates one cell or a few cells originated from one cell, from one stem cell, he was able to make this organelle that looks and kind of functions very much like the villus crypt structure. So basically, he put the isolated cells, stem cells, into culture, and with defined condition, he was able to make a small organoid like the structure present in vivo. So in this culture, he put Wnt agonist, R-spontin, EGF notch ligand, and BMP inhibitor noggin. These are several growth factors he put into culture, and later on you will learn why he put this different growth factors there. But with that and with the stem cells, he was able to recreate the structure, as shown here. This part is crypt-like, right? This is crypt-like. Well, the sum of the part is villus-like. The crypt will have stem cells and transient amplifying cells, and then between this crypt structure, that should be the villus structure. And then when he cut out each part from this kind of structure cell culture, he was able to prove that those are indeed villus-like domain and as well as the crypt-like domain. And he showed that by green fluorescence labeling, those green fluorescence labeling basically is try to visualize the Paneth cell, because the Paneth cell will secrete lysozyme. So they will secrete lysozyme. If you have a substrate of lysozyme, you can make that part to glue green fluorescence. So the green fluorescence there demonstrates the presence of Paneth cell there, while the red fluorescence is the antibody staining of actin, of F-actin. The F-actin is mainly found at the apex of those, villus-like domain. So at the apex, usually you find F-actin positivity. So that basically illustrates the organization of different cells within that organelle structure. And now, at the beginning, I told you there were two theories, two models there. And now here I want to give you a current model that integrates the two models. So basically, they say at the bottom, you have a kind of stem cell. At the +4 position, you have another kind of stem cell. They're kind of distinctive from each other, although they're related to each other. So they say this crypt-based columnar cell, this CBC stem cell, I wouldn't touch these markers. They bear similar markers and unique markers. But the special thing of this columnar stem cell are they are proliferative. They self-renew very, very actively, but they're also damage sensitive. That means if you apply some kind of damage like irradiation, you can eliminate these cells at the very bottom of this crypt. Well, the +4 cells, they are kind of quiescent. They're not very highly proliferative, not as proliferative as the cells at the bottom of the crypt. And they are damage resistant. So if you have some toxin that that can eliminate the stem cell, and these cells at the 4+4 position will be more resistant. So we consider that the +4 cell is the reserve stem cells. Well, the CBC cell, the kilometer cells are the functional stem cells. That's the current view of how things work. So, and as well, that also put a hierarchy into this new model. Basically, this LGR5 positive kilometer cell will give rise to the +4 stem cell. So the +4 stem cell now will serve as a reserve, but originally they were from the kilometer cells. So that's the current model of how things work in the small intestine. But here I want to point to you another thing that's very, very interesting. That's like a couple of years after they did the linear tracking to show this to finally put a lid on that discussion. What is the stem cell in there? They did a linear tracking of LGR5 positive cells, but just a little bit later, scientists demonstrate this concept called neutral drift. Let me explain to you what happened, but I hope you can envision the breeding strategy there to give rise to this kind of animal for the scientists to do the study. So basically, the scientists were able to use LGR5 Cree with another animal, which is I would say it's sometimes called confetti animal, or rainbow animal. So by that crossing, so they can label the cells, the individual cells, with different color, and that labeling is random. It's not like they pick a cell to label as red and then pick another cell to label as green. Since doesn't work like that. The labeling itself is random. That means as long as the cell is LGR5 positive, they just randomly pick a color. We had the discussion about this kind of labeling in manipulating mouse embryo. Okay, I hope you get back to that and study this strategy. As I said, it's going to be a concept I emphasize a lot. So by this technique, they label the stem cells at the base, the kilometer cells, with different colors. But within 2 weeks, you're going to see that the lining now become rainbow-like. Why is rainbow-like? Because the stem cell at the beginning have different colors. Now, each of the stem cell will contribute to cell renewal, cell replacement. That's why you see rainbow color of the epithelium. But later on, at 6 weeks, you start to observe that one of the colors starts to dominate, and some others, the colors start to disappear. And at 6 months, the entire lining is now red. As I said, this is a random process. Don't get me wrong that they are going to be red only. They could be green dominant. The entire lining now become green. That's a possibility there. So the concept here is now one population of cell will dominate and give rise to the entire lining. So that concept is called neutral drift. In that concept, it says that the stem cells, the kilometer cells, don't behave as an individual, they behave like a group. So some of the cells will immediately to become transient amplifying cells. They don't self-renew. Therefore, in this symmetrical division of some of the cells, you have the resulting of the initial cell division is 2 transient amplifying cells. You lose the stem cell population, not the population. You lose that individual stem cell. Okay, but some of those will keep the stem cell identity by giving rise to 2 kilometer cells. So that means you don't have any transient amplifying cell. So the cell in that crypt base, they behave differently. Some will go directly to differentiation, while some will keep the identity. Which one will keep? It's mystical. Maybe that process is random. But you have this important concept here called neutral drift that says in a lot of adult stem cell niche, the cells behave like a group, like a population, not as an individual. So you can't predict those cells. Every one of those cells will become differentiated and become stem cells at the same time. That's the definition of that, right? The stem cell will self-renew through a symmetrical division, while some other cells will go down differentiation. So that's the concept we learned. But if you consider the stem cells in that place as a population, they still fulfill the same task. But just some cells will go down to get differentiated, and some others will keep the stem cell identity. And the concept here is called neutral drift. So the important thing for me is you must appreciate how the experiment is done. This is one of the most important concepts in my class. Is how you do a linear tracking using the creative tools we have introduced to you. So this is I would say this is the last slide. We still have one more, but you can study that one. It's not so important. It's about the stem cells in the large intestine. So here, this slide describes the signaling transduction between the two cell types at the base of the crypt. The one is the Paneth cell, that's the supporting cell, right? The niche cell. The other is the LGR5 positive columnar cell, the crypt base columnar stem cell. So the signal transduction here is this Paneth cell will secrete EGF, will secrete Wnt and into the intracellular, extracellular domain. And now the recipient cell, that's the LGR5 positive stem cell, now will get the stimuli from EGF and from Wnt. And then the Paneth cell will also give rise to the R-spondin. The R-spondin is a cofactor for the Wnt pathway. So for the Wnt pathway to get completely activated, it needs Wnt, but also a helper that is R-spondin. Then with that, the LGR5 is a part of the receptor. Okay, the LGR5 itself is a part of the receptor. This is the marker of the stem cells because this component of the Wnt pathway is unique for the stem cells here. And the function of LGR5 is to fully activate the Wnt pathway. So this is just a co-receptor here responding to R-spondin. And therefore, when Wnt binds to the ligand complex, it's going to stabilize beta-catenin, and then beta-catenin will move into the nucleus and then be associated with TCF4 and then causing transcription that maintains stemness. The EGF factor will go through a MAPK pathway through a cascade of phosphorylation and also does the same thing to maintain stemness. And same for the Notch signaling pathway. But BMP, through a ligand, the BMP receptor, will play a negative regulatory role. Therefore, we remember there are 4 pathways for this for the regulation between the Paneth cell and the stem cell. But the important thing is once you learn this, you start to appreciate the why in this paper, in Hans Clever's paper, they used the Wnt agonist R-spondin EGF Notch ligand, but an inhibitor of BMP4 in this culture condition, because he wants to simulate the environment that promotes the interaction between Paneth cell and the stem cell, and that will also promote the differentiation of the stem cell. Okay, a lot of work was done by Hans Clever there. So that's the end of today's lecture. If you have a question, you can stay behind. Otherwise, you can leave. Thank you.